Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Heather Bruce, and I'm a postdoc at the Marine Biological Laboratory. I will be moderating today with Eyal Karzbrun, a postdoc at UC Santa Barbara. We are excited to highlight the work of our outstanding postdoctoral members. Today, Chi-Chung Wu from the Max Planck Institute for Heart and Lung Research and Isabel Espinosa Medina with HHMI at Janelia Research Campus will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. So please enter your questions in the Zoom Q&A box. Chi-Chung Wu received his Bachelor of Science in Molecular Biotechnology at the Chinese University of Hong Kong. He joined the International Max Planck Research School for Cell Developmental and Systems Biology in Dresden, Germany, for his doctoral studies under the supervision of Professor Gilbert Weidinger to study heart regeneration in zebrafish. Here, Chi-Chung Wu received his PhD in 2016. Currently, he is a postdoctoral fellow in the laboratory of Professor Didier Stanier in the Max Planck Institute of Heart and Lung Research. So please take it away, Chi-Chung Wu.
Okay, so thank you very much for the kind introduction and also the opportunity to actually present my work and share my work today with all of you. And so my main scientific interest is actually to understand how during embryonic to postnatal heart development, the cardiomyocytes, which is the contractile unit of the heart, go from diploid to become polyploid, and potentially also try to understand why this happens at all. And so such transition from diploid to polyploid in cardiomyocytes is actually an example of somatic polyploidization, meaning the polyploidization, this process actually happens during the life cycle of a somatic cells. And so this is basically non-inheritable, unlike the whole genome duplication events that we have seen during evolution. And of course, polyploidy is not just simply multiplying the genome. So it has been implicated in a wide range of biological processes in different species during the development, tissue repair, or age-associated disease like cancer as well. And so polyploidization is usually the result of cell cycle variants. And so here you're looking at basically a typical cell division, a cell cycle. So a cell will enter a G1 and then goes through S, G2, and M phase, and in the end, they will divide into two daughter cells. So a cell can become polyploid by endocycling, meaning they bypass M phase and oscillate between G and S phases. And after each round, basically the genome will be duplicated. And another way to become polyploid is actually by endomitosis, meaning the cell will actually enter M phase. However, it will fail at some point. And so depending on where it failed, when it failed, you will have different outcome. And so if karyokinesis is a factor, meaning the segregation of the chromosomes, then you end up with a single polyploid nucleus. However, if cytokinesis is a factor, and then you end up with a cell with multiple diploid or polyploid nuclei. And so coming back to the cell type of interest, cardiomyocytes. So this is not a unique, polyploid is not a unique feature to mouse or rat cardiomyocytes. It is actually quite prevalent in different mammalian species, including pigs and human as well, unlike the lower vertebrates, in contrast to the lower vertebrates like zebrafish and newt. And in fact, mammalian cardiomyocytes or rodent cardiomyocytes, they become the polyploidization process is actually developmentally regulated. And so if you were to follow the cardiomyocytes from different embryonic to postnatal stages, you will find that in the embryonic heart, they're basically predominantly mononuclear and diploid, which is consistent with the fact that they're very efficient in undergoing cell division, as shown here. However, shortly after birth, starting from day 3 or day 4, they start to lose their ability to complete cytokinesis. And so instead of cell division, they basically become multinucleated or binucleated. Or in some other cases, they will become mononuclear polyploid due to karyokinesis defects. And so as you can see here, basically within the first 2 or 3 weeks of age, almost more than 90% of all cardiomyocytes in the mouse heart would have become polyploid. And more recently, it has also been shown that instead of cell cycle variants, cell fusion might also appears to contribute to this polyploidization process. And so cardiomyocyte polyploidy has actually been reported for more than 2 decades ago. However, until now, we still know relatively little about the physiological significance of this process. Recent studies, however, from Henry Sukoff lab and also the Burns lab, they have shown very nicely in both zebrafish and mouse that there's a very strong negative correlation between the degree of polyploidization and the proliferative and regenerative capacity of the heart. And so understanding how cardiomyocytes become polyploid would have broad relevance basically to both developmental biology and potentially also regenerative medicine as well. And so the main question that I want to answer since the start of my postdoc is actually what prevents postnatal cardiomyocytes from dividing. And so if you think about the regulation of cardiomyocyte cytokinesis, of course, it's very intuitive to think that there will be a cell autonomous component, for example, the cell cycle machinery or some sort of signaling pathways that's changed after birth, because at the end of the day, it is the cardiomyocytes that become polyploid. And in fact, several of such factors have been shown in the past few years to be important, like AT2, a very critical component of cell cycle, or thyroid hormone signaling, or TNI3K kinase, which has been shown to be also important factors modulating cardiomyocyte cell cycle and cytokinesis as well, just to name a few here. However, in these projects, we are more interested in understanding how the environment of the heart, of the postnatal heart, actually influence this process, because obviously cardiomyocytes is not the only cell types in the heart. And in particular, we are interested in the role of fibroblast and also the ECM in this polyploidization or binucleation process. And so to address this question, we decided to use primary postnatal cardiomyocyte culture as our model, because the main reason for that is we can basically test one variable at a time, because there are a lot of interaction between different cell types and the ECM and so on in vivo, which makes it very difficult to tease out the individual kind of contribution to polyploidization. And so at the beginning of the project, we established a EDU pulse chase-based assay to study cardiomyocyte cytokinesis. And so the idea is pretty simple. We cultured the cardiomyocytes. We gave them a short EDU pulse to mark the cycling population as shown in green here. And then we continue to culture them for another 18 hours or so to allow them to complete at least one cell cycle. And so at the end, we will quantify the percentage of binuclear cardiomyocytes within the EDU positive population as a readout for successful cytokinesis. And so here's how it actually looks like. So if you culture embryonic cardiomyocytes, so you will see that the majority of the EDU positive cardiomyocytes are mononucleated and usually comes in paired, suggestive of a very recent cell division event. However, if you culture postnatal cardiomyocytes, you will start to see a lot more of this multinucleated EDU positive chased cardiomyocytes. And so with this assay, we first decided to ask whether postnatal cardiac fibroblasts actually play a role in this process. And so what we did was we isolated cardiomyocytes and cardiac fibroblasts from the same heart, and then we basically mixed them together in a dish in different ratios. And what we found was that the more fibroblasts there are in a dish, the more binucleation happened in the cardiomyocytes, suggesting that fibroblasts is influencing as cardiomyocyte cytokinesis. And these results are basically in line with a publication from last year showing that the ablation of a subpopulation of cardiac fibroblasts in vivo actually reduces postnatal myocyte polypoidization. And so after that, we wanted to understand what is the role of the ECM, because secretion or maintenance of the ECM is one of the major functions of fibroblast. And so for that, we decided to go for a cell-derived ECM approach. And it's actually pretty straightforward. So we basically isolated cardiac fibroblasts from different stages, like from embryonic and postnatal stages, and then we culture them for an extended period of time, roughly 2 weeks, and during which these fibroblasts, they will produce and secrete the ECM proteins, which will then be deposited onto the culture dish, the surface of the culture dish. And so after decellularization, we can use the remaining matrices for cardiomyocyte culture. And so to cut the long story short, basically the main findings from these experiments was that irrespective of the age of cardiomyocytes that you cultured, embryonic or postnatal, they always binucleate more when they are on postnatal ECM as opposed to embryonic cardiac fibroblast derived ECM, suggesting that the postnatal ECM provides a non-permissive environment for cardiomyocyte cytokinesis. And so, of course, these experiments or these results also allows us to basically gave us an opportunity to try to identify some specific ECM components that could be pro-cytokinesis. And so for that, we basically subjected these two matrices for mass spec analysis. And here we are focusing on the embryonic enriched ECM proteins, which are potentially pro-cytokinesis. And we cross-reference this with the gene expression data, and we only select those ECM proteins whose expression mRNA level actually goes down in during the first week of birth. And that's where led us to the findings that two ECM proteins, nephronectin and SLIT2, both are able to promote cardiomyocyte cytokinesis in vitro. And so we were pretty excited about this, because also puzzled, because like sleep robosignaling, for instance, has been studied for quite extensively in the context of axon guidance and also embryonic heart development, but not really postnatal or cytokinesis, not normally related to cytokinesis. And so we decided to dig a little deeper to follow on the role of sleep robosignaling. And so the first question we ask is, where is SLIT2 expressed? Of course, from our ECM experiments, we know that SLIT2 is expressed and secreted by fibroblasts. But when we analyzed other published data sets of sorted cells from the postnatal heart, we found that SLIT ligands and receptors are actually expressed in multiple cell types, including cardiomyocytes. And we can also confirm that by immunostaining of P1 isolated mouse cardiomyocytes. And we found that expression can be detected in both non-myocytes and myocytes. But what is surprising to us is that there's actually a lot of pretty strong nuclear localization of the protein in cardiomyocytes, which is quite different from what one would normally think SLIT2 would be secreted once they're expressed. And so we thought, okay, maybe this suggests that there might be some sort of functions in cardiomyocytes themselves. And so we went on and checked other data sets as well. So here is a single nuclei RNA-seq of cardiomyocytes generated in Eric Olsen's lab recently, last year. And so here we found that SLIT2 and the receptors ROBO1 and 2 appears to be enriched or expressed in a subpopulation of cardiomyocytes, early postnatal cardiomyocytes, P2 and P4 as shown here. But of course, the question is which kind of cardiomyocytes. And so we look at another data set from Ed Maurice's lab, where they sorted mono and binuclear cardiomyocytes for RNA-seq. And from our analysis, we found that both ligands, SLIT2 and 3, and also the receptor ROBO1 are significantly enriched in a mononuclear population compared to the binuclear counterparts, again, suggesting that there might be some cell autonomous function in cardiomyocytes, controlling the cytokinesis or polypolarization. And of course, the key question then is, do they actually have a function or not, these ligands? And so for that, we went back to our binucleation assay, which I've shown you before in vitro. And here we use siRNA to knock down SLIT2 and SLIT3. And so as you can see here, compared to control, SLIT2 and SLIT3 loss of function or knockdown significantly increased the percentage of binuclear EDU-positive cardiomyocytes, suggesting that both ligands are important or are required for cytokinesis. And then the next question is, so the ligand seems to be required, but which part of the ligand, of the protein? And the reason I'm saying that is because we know that from literature, SLIT proteins are post-translationally modified, meaning they are actually proteolytically cleaved around this site, which will then result in two different fragments, the bigger N-terminal fragments, which contains all these LRR repeats, important for binding to roboreceptors, and also a shorter C-terminal fragments, which cannot bind to roboreceptors, but other receptors. And so to test which one is actually important, we basically repeated our siRNA experiment. We again compared to control, SLIT2 knockdown, increases binucleation. And so we try to see if we put back the recombinant protein N and C-terminal fragment, we try to understand which one of them can actually rescue the phenotype. And in fact, what we found was that both fragments, N-terminal or C-terminal fragments, are both capable of rescuing the phenotype, suggesting that potentially both robo-dependent and robo-independent signaling cascade or events are important or involved in the function of SLIT2 on cardiomyocyte cytokinesis. And so, of course, the question that many people then ask, does it work also in vivo as well? So it looks great in vitro, but what about in vivo? And so for that, we performed a recombinant protein injection experiment. So here we took pups and then we injected N-terminal fragments of SLIT2 recombinant protein daily from P1 to P4. And then on P3, we gave a single EDU injection, and then we harvested the heart at P5. And after dissociation, we quantified the amount or the percentage of EDU-positive cardiomyocytes, whether they're mononuclear or binuclear. So basically an in vivo version of the binucleation assay. And so from these experiments, what we found was that compared to PBS injected controls, if we inject SLIT2, there are significantly higher percentage of mononuclear EDU-positive cardiomyocytes in the heart, suggesting that SLIT2 could promote cardiomyocyte cytokinesis in vivo as well. And so then, of course, the next question is obviously what could be the potential underlying molecular mechanisms. And so for that, we have been working on both gain of function and loss of function of RNA-seq and so on. And but today I'm only going to talk about the mass spec analysis, the gain of function experiments we did. So we basically, what we did was we treated postnatal cardiomyocytes in vitro with SLIT2 and nephronectin recombinant proteins. So nephronectin is another ECM protein that we identified from our mass spec analysis, which I've shown you before. And here we are basically looking for commonly regulated proteins enriched or repressed after treating them with these proteins. And these experiments basically led us to one of the candidates that we are working on now, and that is the HNRNPC. So HNRNPC is an RNA binding protein, which is important for RNA metabolism, for instance, the stability, splicing, and so on and so forth. And in the context of cancer cell lines, it has been actually shown that the overexpression of this gene or this protein would lead to the reduction of translation of ROB kinase. And of course, RB kinase is one of the very important critical protein for mitosis and cytokinesis as well. And so we decided to do some experiments on this gene, on this protein. And so the first experiment we did, again, we went back to our binucleation assay. We tried to knock it down, this HNRNPC gene. And what we found was that compared to control, there's a significant reduction in binucleation of cardiomyocytes in the loss of function, suggesting that this RNA binding protein is normally act as an inhibitor of cytokinesis. And so to test whether this protein or this gene actually could potentially function downstream of SLIT2. And so we would perform another round of experiment where we knock down SLIT2, again, compared to control, more binucleation. And so in another condition, we basically knock down this RNA binding protein together on top of SLIT2. And here you can see that basically there's a pretty strong rescue of the phenotype, even lower to the level that is even lower than control, suggesting that at least partly this HNRNPC, this RNA binding protein is working downstream of SLIT2. Okay, so in summary, coming back to the original question, what prevents postnatal cardiomyocytes from dividing? So I hope I've shown you today that postnatal ECM is playing a role by providing a non-permissive environment for cytokinesis. And SLIT ligands are actually some of the candidates that we found, and potentially SLIT2 potentially promote cytokinesis by reducing the level of the RNA binding protein HNRNPC. And so, right, okay, so I'm just going to end my talk here by the acknowledge of thanking people, everyone who are involved in this project, of course, the STANI lab members and different people from different facilities in the Institute, our collaborators from Oxford, our funding sources, and of course, all of you for your attention. So I'm happy to take questions. Thank you very much.
Thank you, Chi-Chung. That was fantastic. So now we can take questions. You can type them in the chat in the Q&A. So I was wondering, I was wondering if there's any diseases that affect heart cell binucleation, like do they reduce binucleation or increase it? And then what would the effect be?
So it has been reported that there's some congenital heart diseases would change the number or the percentage of multinuclear cardiomyocytes in infants. So that has been shown to have something to do with like the expression of ACT2 and so on. So yeah, some congenital heart diseases. Yeah.
And an anonymous attendee asks, what do you think SLIT2 is doing in the nucleus?
Yeah, that's a very good question, which we don't know. I mean, SLIT2 is not really known for its role as a transcription factor, for instance. But so we're working on that. But it seems like, because like HNRNPC, one of the target that we found, it seems like it's RNA binding protein, which is also primarily localized in the nucleus. So perhaps there's some sort of protein-protein interaction happening between SLIT2 and other proteins in the nucleus. But yeah, we're working on that. I will be happy to let you know once we find something.
And Stephanie Wu asks, do SLIT ligands show nuclear localization in other cell types, for example, neurons?
So I think in neurons, it's secreted. So it has been shown in pancreas. I think it's SLIT3, not SLIT2. That is localized, nuclear localized. But it's yeah. So, but normally people would, associate SLIT protein to be secreted. Yeah.
Ramin Yadigari asks, any indication of dysregulation of SLT2 or 3 ligands and other ECM-related genes in human cardiovascular diseases?
Human cardiovascular diseases. I haven't really looked into that, especially not human. But in mouse, what we found was actually that after injury, for instance, like some SLT and ROBO expressions are actually upregulated or downregulated depending on the age and so on. So there might be some correlation with the regeneration, but it's not entirely clear at the moment.
And Augusto Ortega Granillo asks or says, cool talk. A naive question, once cardiomyocytes are binucleated later in development, do you think it would be possible to induce cytokinesis to go back to diploidy?
Yeah, that's a very good question. So, so like in liver, it has the hepatocytes, they also polyploid, and has been shown that there's potentially they can actually enter cell cycle again, and some of them will be turning back to diploid. It has been shown before. But in cardiomyocytes, the general understanding or belief is that once they become polyploid, they seem to become post-mitotic. And I know that there are people out there also trying to see if one can actually force this polyploid cardiomyocytes or multinucleated cardiomyocytes to re-enter cell cycle. But at least from what I know, from what I understand, the mononuclear population are much more responsive, like to injury or to stimuli, mitogens and so on. So, but it would be cool if one could do that.
And Peng Huang asks, is SLT2 required in fibroblasts or cardiomyocytes?
Oh yeah, so we don't know. The short answer is we don't know. So we are currently trying to do some mouse work, basically knock out these genes in different cell types, fibroblasts, cardiomyocytes, whatnot, and see, where, which source it's more important. But based on the expression level, fibroblasts has a much higher expression level compared to cardiomyocytes. So I would not be surprised that they're more, just knocking it out from cardiomyocytes might not see a phenotype because there's so much more secreted by fibroblasts. Yeah.
And another question from Augusto Ortega Granillo is, in your SLT2 shRNA experiments in binucleated assay, are you culturing fibroblasts as well? It wasn't clear to me as you were knocking down SLT2 and fibroblasts or cardiomyocytes are synthesizing SLT2 as well.
Right. So actually that's a good question. So basically when what I referred, so when we're doing cardiomyocyte culture, we try to purify it as much as possible. But at the end of the day, the best we could achieve routinely is around, I would say, 90% to 90-something%. So there will always be some other cell types in there. And but in those experiments, when I do siRNA experiments, those are actually 90% to 90-something% pure cardiomyocytes. So of course, I cannot say for sure that what I'm seeing has nothing to do with other cell types that potentially are expressing SLT2 in the dish, but the majority of the cells that we are looking at there are cardiomyocytes. Yeah.
Mathilde Cooley asks, did you look at the expression of ROBO in your single nuclear RNA?
Right, you totally got me here. So yeah, I actually, so we have done that experiments very recently, and the findings that, so the results that we found was that actually surprisingly, when we knock down robots in cardiomyocytes with those binucleation assay, we found a complete opposite effect as to knocking down SLT2. So knocking down SLT2, you see more binucleation. So you would expect if you knock down robo, you would see similar phenotypes. But not only do we not see that, we actually see as opposite phenotype. It seems to promote cytokinesis. So we are still trying to understand what is going on there. So, but it's a very good question. Thank you.
Okay, so we can move on to the next speaker now. Thank you, Chi-Chung.
Thank you again.
Thank you, Heather and Chi-Chung. My name is Eyal Karzbrun, and I'm a postdoc at UCSB, and I will be the moderator for the second talk today. I have the pleasure to introduce Dr. Isabel Espinosa Medina. Isabel is a postdoctoral fellow at Janelia Research Campus, where she is working with Dr. Lee and Dr. Koyama. Isabel has an exceptionally strong scientific track record. She has started her way at the Autónoma University of Madrid, Spain, where she earned her bachelor degree in biology and biochemistry. She then did a master's degree in neurobiology and molecular biomedicine, where she was first introduced to the beautiful world of developmental biology and became interested in how cells give rise to organs. After a successful master's degree, Isabel moved to Paris to do her PhD at École Normale Supérieure. During her PhD, Isabel discovered the molecular mechanisms by which progenitor cells in the periphery nervous systems migrate towards their final position. This significant work was published in 2 science articles. The importance and challenge in tracking individual cells during embryonic development has motivated Isabel to develop new imaging-based genetic tools that allow recording of cell histories in zebrafish and mouse, which is now pursuing in Janelia. Isabel is the recipient of multiple distinguished awards, including the NIH Embryology Scholarship and the EMBO Fellowship. I'm very excited to hear what Isabel has for us today. Isabel, the floor is yours.
Hello, thank you very much for that kind introduction. And thank you for the organizers for inviting me to this seminar series, which so far has been really amazing. And I congratulate the previous speaker for a really good talk too. So now I'm going to share my screen, if it allows me. Sorry, I cannot see my screen for some reason. There you go. All right. Can you all see it? Okay. So today I want to introduce a new tool I developed based on CRISPR-Cas9 for temporal labeling and manipulation of vertebrate cell lineages. And as Eyal just said, the main interest during my whole career is to actually understand how complex organisms, how complex and diverse tissues and organs form from single cells. And understanding this, resolving this fundamental biological question would allow us to understand how cells originate, how they interact, and also the molecular factors that allow them to become what they finally become within the tissues. And just by reconstructing and labeling cell lineages, we actually cannot resolve this question. We also need to be able to label and manipulate those cells sequentially to be able to solve the molecular mechanisms involved. And so we need genetic tools with temporal control. Current tools for lineage tracing unfortunately lack temporal resolution. So classical tools are based on the recombination of a label, for instance here YFP label by a Cre recombinase that labels a progenitor cell and all the progeny with the same color in the spatial context. But these techniques do not reveal temporal information. You cannot distinguish between subsequent generations of cells. By contrast, sequencing-based techniques allow us to figure out the cell-specific type of interest, but because we need to perform tissue dissociation, we lose crucial spatial and temporal information. So during my postdoc, I have developed a new tool to solve these limitations, which is called tempo and allow us to subsequently label different generation of cells with different colors. And also this way we can genetically access different generations of cells in vertebrates, both in mouse and zebrafish. This tool is based on CRISPR-Cas9 instead of in recombinases because of the more unlimited potential of this tool. And especially because in the lab, we recently developed another type of genetic switches that we call CASA switches, in which fluorescent reporters are disrupted by a target sequence for a guide RNA. And that target sequence is in between two direct repeats of that fluorophore that is disrupted. After CRISPR-Cas9 double-strand break into that in the middle of those repeats and single-strand annealing repair, this is a repair mechanism that is very conserved in evolution. Then after that process, we can have an active fluorescent reporter. So based on this, I'm going to tell you how I built tempo. So tempo is a compact transgene formed by three reporters, which are expressed in different coding frames. At first, only the CFP fluorescent protein here at the end of the cistron is expressed, so all the cells will be expressed in CFP. Subsequent cuts and repairs by single-strand annealing will reveal a cascade of fluorescent reporters after CFP, RFP, and then YFP. But how can we make this happen in this order? Well, because in addition to that transgene, we have also a conditional guide RNA cascade that we express. Guide RNA 1 is able, along with Cas9, to repair RFP, and in parallel, repair a conditional guide RNA 2, which then subsequently can help the repair of the last reporter. Importantly, this compact system allows to integrate these transgenes into vertebrate systems, including mouse and zebrafish, as I will show in a second. It works only in this predefined order. Each protein is expressed at one time. The system is irreversible, and when one turns on, the previously one turns off, and only can be activated by this type of repair mechanism. And very importantly, it only occurs in dividing cells. All right, so let me show you how this works in zebrafish. We first introduced this tempo and Cas9 constructs, including the guide RNAs, under an ubiquitous promoter into one cell stage zebrafish embryos, and then imaged consecutive stages of development of the zebrafish embryos to test the progression of the tempo cascade. What we found is that at already 8 hours post-fertilization in the fish injected, most cells are CFP positive, and as expected, at 24 hours, most cells have turned on the second color, RFP. And 24 hours later, we see already the appearance of the third color that becomes predominant at 72 and 96 hours. Interestingly, in this clone that I'm showing you here, cells continue to divide and express the last color of the cascade, while those cells labeled A here that did not divide in this time frame, they cannot transition in the cascade as expected because this mechanism depends on cell division. As you can see, the color transitions happen in the predefined order, and this system is also very efficient as the transitions have an efficiency of higher than 90 or 80% for both color transitions. We have now generated transgenic zebrafish lines that express tempo under the conditional promoter UAS so that we can activate this system in specific lineages of our interest. And as proof of concept, I'm going to show you the activation of tempo in a particular neuronal lineage of the zebrafish hindbrain that in addition, we have been studied with Minoru Koyama in the lab. So this is the VS62 hindbrain domain in which interneurons that are very important for locomotion in zebrafish are distributed in a birth-dependent way. So those V2A interneurons that are born early, they distribute in a more lateral position of the hindbrain and are involved in early responses of the zebrafish, like escape, struggle, while those V2A interneurons that are born later occupy more middle position, closer to the midline in the hindbrain, and also dorsally, they are dorsally stacked to those born earlier. These later neurons are involved in more complex behaviors, including exploratory behaviors in the fish. So to test whether tempo works in this lineage as well, we generated transgenic conditional lines, as I mentioned, a UAS tempo line that driven by the VS62 GAL4 transgene can be expressed only in VS2 interneurons, and a HER4.1 Cas9 along with an H2B halo tag label in the nuclei that is expressed only in neuron progenitors. So the results by crossing these two lines in these zebrafish larvae at 6 days post-fertilization show that the tempo label neurons are distributed with respect to their birth date as expected. So those neurons born earlier distribute more laterally, while those that are born later distribute more medially. While with respect to the dorsoventral axis, we can see that those born later stack dorsally to the earlier born neurons, but in a rhombomer 6, by contrast, we see the opposite. But this is also expected because in that particular rhombomer, later born neurons are known to migrate, as you can see in this cartoon, later stage, to migrate ventrally to the earlier born one. So tempo recapitulates also this pattern of distribution of birth-related neurons. And we also found that the projections of the latest born neurons are more lateral to those born earlier. And this was also found in the previous studies. But in addition to the previous studies, tempo reveals a higher level of resolution of temporality because we have more labels in the same sample. And we now have genetic access to these populations that previously couldn't be accessed with previous techniques. But not only we can use this type of tool in transparent models in which we can actually track development life, but it would be really useful to have tempo implemented in other models which do not have the such an easy way to be imaged along development. And I'm talking about mouse. So it would be really important to adapt tempo to mouse to retrospectively track cell histories. So how did we do it? Well, we adapted the previous constructs I showed you and electroporated them at 12 days of development into the brain of mouse embryos, and then imaged brain samples at consecutive developmental stages. And this way we targeted the mouse cortex because this is a perfect setting as proof of concept, because it is known that the mouse cortex develops in an inside-out way. So basically, earlier born neurons, here represented in blue, occupy the lowest layers of the cortex, while progenitors that are born later, they produce neurons that migrate past the earlier born ones and occupy upper layers. And the latest born progenitors in this case will produce glia astrocytes after neurogenesis is complete. So how did tempo behave when we did these experiments? Well, we found that tempo actually follows the predefined order as expected. First, CFP progenitors that migrate are generated. 1 day later, we start to see progenitors that are RFP and then continue to migrate to the cortical plate, while the latest one to appear are YFP. And this is also observed in this plot of the ventricular zone. And this pattern of inside-out generation of neurons is also observed in postnatal brains labeled by tempo. So we confirm that tempo recapitulates the inside-out pattern of layer formation, and also the neuron to glia distribution, as latest born cells labeled YFP mainly contribute to astrocytes. And also in correlation with previous work that has shown that astrocytes birth date do not correlate with layer distribution, we also didn't find any correlation. Interestingly, we can electroporate tempo at different stages of development and thus target different populations of progenitors and characterize then corticogenesis from lower layers if we electroporate at 12.5, or starting at upper layers at 14.5, or even characterize more gliogenesis states after electroporation at 16.5. And results by doing this type of analysis have revealed that astrocytes that form the most superficial layer of the cortex, those astrocytes actually have a lower progression of the cascade, as you can see in this plot, when we electroporate the brain at 16.5 compared to earlier stages. This suggests that L1 layer astrocytes are being mainly generated just after 16.5, and probably they don't have many more cell divisions left after this stage of development. And this is in contrast to some other studies that claim that L1 astrocytes could be generated both at perinatal and postnatal stages. But in addition to labeling consecutive cell generations with tempo, we figured that it would be really interesting to use it to manipulate cascades of transcription factors, because during development, genetic cascades are essential. For instance, here I'm showing the example of the progressive generation of endocrine cells during development, which happens because transcription factors are deployed in a particular order. Or the same happens in the central nervous system of mammals. If the sequence of transcription factors does not occur in that way, we wouldn't have the same neuronal types forming the central nervous system. So there's no tool currently in vertebrates allowing the manipulation of effector genes in a cascade in a predefined order. So what we did was to adapt tempo for this purpose, and we coupled the expression of effector genes with that of the fluorescent reporters in different temporal windows. As you can see here, in the temporal window 1, you have a gene of interest here, or in the temporal window 2, the gene is just next to RFP. And in collaboration with Daniel Feliciano in the lab, what we decided was to try to manipulate the spatiotemporal distribution of glia and neurons in the mouse cortex by manipulating cell cycle regulators, cyclin D1 or cyclin B1, that have been correlated with progenitor commitment or progenitor stem cell maintenance, respectively. And so first, by overexpressing cyclin D1, which has been implicated in progenitor differentiation, by overexpressing it in the first or second temporal window, what we obtain is a shift in the lineage commitment towards lower layer neurons. As you can see, that not only affects the temporal window manipulated, but also the subsequent temporal windows. And this is most apparent in the manipulation of the second temporal window, in which many cells that in the control sample will mainly produce upper layer neurons now produce lower layer neurons at the expense of upper layer neurons. By contrast, overexpression of cyclin B1 in different temporal windows led to an increase in astrocyte numbers only in the temporal window of interest that we manipulated, but not in the other ones. And this was expected in the sense that cyclin B1 has been correlated with progenitor maintenance. So what is happening here is that it's delaying the fate towards a later fate or astrocytes. So here we show that tempo can be used not only for labeling consecutive cell generations, but also to deploy effector genes, and thus to study the effect of molecular factors in the cell specification cascade, and also both cell autonomous and non-cell autonomous effects, because we can visualize both the perturbed and non-perturbed populations simultaneously. But of course, we can still improve our techniques, and tempo has temporal resolution, as its name indicates, but it doesn't provide clonal resolution. So if we would want to track multiple cell lineages in parallel, we will need to add another layer that is clonal resolution. So how to do that? Well, many of you might be familiarized with this technique, Rainbow, which labels multiple progenitors in parallel with different color hues. but doesn't have temporal resolution. We cannot distinguish subsequent cell generations. Well, what I did very recently was to combine both tempo and brainbow in one tool called TempoBow to have both temporal resolution and clonal resolution. How? Well, briefly by instead of deploying a cascade of colors as previously shown, I deploy a cascade of recombinases. The first recombinase, CRE, will activate multiple color hues in the cytoplasm of all these different progenitors. Subsequently, tempo activation of a flippase recombinase will produce in turn the activation of multiple color hues in the nucleus of the following generation of cells. And finally, tempo allows the activation of a halo tag protein in the third temporal window that is shown here with an asterisk. And just to give you a brief summary of the very recent results, here is a postnatal mouse brain electroporated with TempoBow constructs. And here the details showing that earlier born neurons in the mouse cortex only express the first color hues only in the cytoplasm. And the next generation of neurons that is present in more upper layers, as you can see here, also have nuclear hues. And finally, the ones that are born even later that produce neurons and glia, also here, have both cytoplasmic, nuclear, and halo tag marks. So we think that this technique, if combined with functional analysis, with effector genes, or with in situ transcriptomics, is actually a very powerful technique to better understand cell dynamics and how the cell specification cascade can contribute to the formation of complex tissues, both in a mouse, but also in other species. So with that, I would like to thank you all very much for your attention and thank all the members of my lab for their support, the Janelia support team, which are amazing. If you ever have the opportunity to join, there's a call right now, so I really suggest that you try it. And also I want to thank my mentors, the Koyama lab, and my collaborators in the Cristina Pujades lab. Thank you.
Thank you, Isabel, for the great talk. Everybody's welcome to upload their questions at the Q&A, and I'll read them. In the meantime, I'll ask a question. Isabel, do you think you can modulate the temporal rate at which tempo works, meaning that you would change fluorescent states every two cell divisions and not just one? And a follow-up question is, are there any errors at that rate? Like if you see a pink cell, could it, or a blue cell, could have been that it has undergone divisions, but somehow the molecular switch did not work?
Definitely, yeah, that's a great question. Actually, right now, our thoughts is that the transition doesn't happen every cell cycle, actually, because CRISPR-Cas9 still has some stochastic activity. So we calculate that it happens every 2 to 3 cell cycles right now. And we are implementing actually some heat shock Cas9 versions, so we can give it a stronger pulse of expression and help speed up the cascade, for instance. We could also want to slow it down, and for that there are guide RNA switches that can be tweaked so that they work more or less fast. But currently the guide RNA switch has an efficiency of around 70%. So definitely making the cascade longer will also lead to more errors. Yeah.
Maybe I'll ask a second question in the meantime. Most of the work that you've shown is focused on the brain. Do you envision moving to other developmental questions?
Yes, actually. So because I'm still in love with the peripheral nervous system and how it coordinates bodily functions, basically, my future studies are going to be focusing on the interaction between the autonomic nervous system and the organs, and then I will use these tempo tools to evaluate how the neurons and the organs form in a coordinated manner. yeah, specifically the pancreas and the nervous system.
I see. Mathilde Julie has a question. Did you try to electroporate the tempo expression plasmid into her 4.1 Cree adult zebrafish brain?
That would be useful. We the tempo constructs we have now are not dependent on Cree for zebrafish, so we would need to just generate that version of tempo constructs. The ones we have right now have here for Cas9 directly expressed, but it would be great to have another layer of control. If you have that line, I can get into contact with you.
Nambora Reye has another question. Can tempo work with low expressed drivers?
Yes, so one of the main limitations definitely is levels of expression of Cas9. The guide RNAs are very highly expressed. So it's good to have some additionally amplification systems, so to say. So if you have a promoter that is not so highly expressed, then I will definitely suggest to express it with the tetracycline activation system or GAL4 to amplify that signal.
We have another anonymous question. Could tempo be applied to eye development?
Yes, I don't know what do you study, but already HER4 is a promoter expressed in the eye. And during my recordings, I have definitely seen very interesting things like that progenitors seem to divide in a different speed in different regions of the eye, and thus the cascade progresses at different timings. So I'm very happy to discuss more if you're interested.
Okay, I think we can now conclude. So thank you, Chi-Chung and Isabel, for your excellent talks. This seminar has been recorded and will be available on the SDB website starting Monday. Please join us for next month's seminar on Friday, November 12, when Maria Saleh from Stanford University and Muhammad Fatullah Simsek from Cincinnati Children's Hospital will present. And thank you all for coming.